Hello, thank you for making time for me today at the n-Lorem committee. It's been a pleasure working with n-Lorem, and I'm sure the other physicians have also said that. I'm an ophthalmologist, but I also want to really acknowledge the families that are here because although this is personalized trial treatment, this is obviously personal for you, and we've really gone together with some of the families that are here in terms of getting through to these treatments. So I really appreciate the time and the support that you guys are giving us. So let me start by talking about ophthalmology and eyes. So I'm going to reorientate you now from what's been talked about before. And the eyes are very different from what we've just been hearing, pediatric neurology. So we're going to talk about saving sight with ASOs. So we've got a picture here of a patient who's had retinitis pigmentosa. And this and other forms of disease can affect so children from very, very young ages, but also adults. And affects approximately 1,000 to 2,500 sort of children and adults. There's 350 genes which affect the eyes, and they can affect the front of the eye, also the nerves at the back of the eye. And what's important is that there's already been some ASO trials. These ASO trials, one of them is already FDA approved, but for an infectious cause, and the other ones are currently in phase 3. And there's been one prior n-Lorem trial, which you'll hear about tomorrow. Now, there's been an increasing problem in terms of treating inherited retinal diseases and genetic eye disease. And that's because the genetics and gene therapy have really been affected. Lots of these trials, as we just heard, have had to choose this primary outcome measure or endpoint, and essentially we're not hitting them. So since 2017, when the first gene therapy was approved for vision for a congenital eye disease, we've not had any further approvals. These trials cost hundreds of millions of dollars to get through, and yet they get to phase 3, and eventually the primary endpoint is not met, whereas the secondary endpoints are met. There's other problems, and that includes the fact that some of these diseases themselves are causing, or some of these treatments are causing diseases as well. And part of the problem is the disease variability that you're hearing here, because this is a personalized trajectory. It's not like you can't clump patients together. And that really identifies why we need to have these rarer and ultra-rare disease trials. So I'm going to teach you a bit about the eye, just to reorientate yourself. This is all that I learned at medical school, which won't take very long for you guys. So if you look at the eye here, for vision, you need to have light going from the front of the eye to the back of the eye. So we were especially looking at the macula here and the optic nerve. These kind of areas are important to get the signal to the brain. And then what we need is for the light to go through to the back of the eye, to the retina, and hit the photoreceptors, which are at the very back of the eye. That signal passes back through the retina before it goes back to the brain. And that's important because if you lose genes or have problems with cells at different points, you can have different kinds of diseases. And that's really what I'm coming to in terms of the different treatment trials that are required for different diseases. But the eye is an attractive target for ASOs. And again, there are a number of reasons for this. One is that it's easily accessible. We can access and give ASOs to the eye by quick injection, which is much different from the so complex and sometimes more painful operations that are required for intrathecal and other organs. 2, you can see the images on the right-hand side, but they show quantifiable imaging, which means that we can follow patients. And lots of patients have good natural history before we treat them. And so on the top is an autofluorescence image, which looks at an area of atrophy in black, which we've encircled. And on the bottom is an AI-guided quantification of the thickness of the retina. The eye is relatively immune privileged, so there's not much of an immune response when you do an injection to the eye, similar to what we've been hearing with the complement and other stories. And that also isolates from systemic spillover. On top of that, the most common side effect, and as well known, is a bit of inflammation that does occur very rarely from cells. And that's quite easily treatable with steroids which are given locally. The small target volume means that only a small amount of drug is required, and enough can be made for the whole lifetime of the patient. And that also allows long treatment intervals because, unlike the CSF, there's not constant recycling. So treatment intervals can be 6 months to a year in some cases. And finally, we have two eyes. So if one eye is treated, disease is generally symmetric, and we can follow the disease course compared to the other eye, which again covers some of the issues which were sort of mentioned before in terms of natural history progression. So let's go on now to just understand. So we talked about the eye and why I think this offers a very good benefit versus the risk that we talked about earlier on again. But here's a real-world example. So this is one of the genes that our center's been looking at, and it's called peripherin 2, or PRPH2. So in a big study across the world, it was noted to be the 4th most common inherited retinal disease. But it's complex, and you can see the images below. You heard Stan talk about the different phenotypes. This gene causes multiple different phenotypes. In fact, a single mutation can cause multiple different phenotypes. And so that really reiterates the point of personalized longitudinal studies. And luckily, we do have that for a lot of our patients. So the gene that the mutation that we're targeting is the 623, and there's less than 30 US cases, which allows it to be eligible for N of 1 studies. So a very brief bit about the science, but really this gene codes for a protein that is found in the outer segments of photoreceptors, and photoreceptors are important because they're the cells that allow us to convert light to electrical signals. So we saw the back of the retina here. So we're looking at this is the photoreceptor, a really long, quite a long cell with these kind of comb-like elements at the top. And on the edge of those comb-like elements are where the PRPH2 is really important in terms of creating this curvature. And if they don't form properly, then the light does not convert to electrical signals. So our patients are 64-year-old patients with confirmed genetics. They initially had symptoms in their 40s, but has recently been noticing a rapid loss of vision in the left eye, which now sees very little in the central area. And rapid loss of vision now on the right eye, which is now making difficulty in seeing, and driving is now stopped. So really having real-world effects, because the central vision is the most important part of our vision. It's what you use to read now, what you use to see. And this is an example. So this is an autofluorescence image. If you're looking here, the black area, again, is the bad area, and that's very close to the very central area, which is just very enclosed to that large ovoid shape that we're seeing here. I'm not sure if you can get this working here. Yeah, so you're seeing the black area, and then this is the very central part of the vision just here. So this is progression in over 4 years. And again, this is the better seeing eye. So you can see that these areas now have merged. So the two separate islands have now started merging, leaving a very small isthmus here of vision in the central area. So really we're trying to race to save vision. We wanted to get some more data on this, and so we had a look. So my other job is in the stem cells, so we grow retinal organoids. And so with funding, we were able to take stem cells and grow mini retina. And this is important because this allows us to understand this disease and to confirm the disease. And essentially what we showed was that when we counted the photoreceptors, this is a normal, so this is the same line from the patient. So we made iPSCs that you heard about earlier on, made retina, and then we corrected it. And the corrected line has photoreceptors, and then the uncorrected line in green is the areas with the photoreceptors, and the diseased line or case line here had a reduced number of photoreceptors. And we're still studying some of this in terms of the mechanism, but essentially we think this is a dominant negative disease. And then we've got these quantifiable measures. So one area is to look at the distance from central vision and the time points, prediction for time point for blindness. And so we have a predicted curve. So the patient has come in every 6 months, so prior to sort of treatment. And you can see that there's a trajectory that's unfortunately going and predicting within 2 to 2.5 years of vision loss. And so what effect will that have? Again, for you guys who are not familiar, so on the left-hand side is a normal image, obviously, and the right-hand side is that central vision loss. So really quite devastating in terms of identifying patients or faces, and identifying things in terms of reading and things like license plates even. So we're going to talk about this study now. So a drug was developed and was based on RNase-based degradation, and it's mutation-specific. And you'll be hearing from Nadina very soon, who will be talking about how these ASOs are designed. 7 mutation-specific ASOs were identified, and these all had a 10-fold greater targeting of the mutant allele rather than the normal allele. GLP toxicity did find some adverse events, but they were, as expected, with inflammation, especially at higher doses. And with the data and the preclinical data that we showed as well, the FDA approved this within 10 days. They have a 30-day time limit. The primary outcome of this trial was obviously to evaluate safety and tolerability. And that was worked out with various different methods, including systemic things, but especially disease-specific kind of things, so including vision. And you heard about the EEG earlier on. That's electroencephalogram. This is the electroretinogram we did to sort of try and check the retinal function, as well as eye exams. And then we had secondary outcome measures, which were also disease-specific, including visual field testing. So as you can see on the right is the kind of injection that we do. And this is something I do in clinic about 20 times a day, so it's very common. Obviously, I was a bit more nervous with this drug because it took so long to develop, but it really wasn't any different in terms of treatment. And the whole process takes less than 5 minutes. So we managed to treat the patient's left eye, the worst seeing eye, in August. So far, we've had no adverse events related to the drug. And we're planning to increase on a dose escalation with a 3-month dose escalation. So every 3 months, starting with the left eye, as you can see on the top, we'll be going to the left eye again, and then if there are no adverse events, we'll go on to doing the right eye and the left eye in a further 3 months, so in February. So, I think this is what this showed, or what we learned anyway. So we're talking about learning sort of things. I think with the right kind of disease, the right background, and also funding as well, we can get from a disease idea for treatment, approval, to actual needle to the eye, in this case, in about 12 to 18 months, if that's done properly. And so really we want to try and accelerate this because there's an opportunity to treat other diseases. So as we were doing this, we had also discovered other patients who also had the same mutation. And so we've got 6 further patients who we've been following up with good natural history, and in fact, just got the paper accepted looking at their natural history, a personalized natural history. And so we're going to be submitting these to n-Lorem. So I think Laurie's going to be busy soon, I think, looking at these patients. And the benefit of this is that we've leveraged the initial funding that was taken for the first toxicity studies and drug development for these other patients. And then what are we doing for the next steps? Well, it took a long time to develop the IRB approval and the contracting. And we've heard already, I think, from Neil Schneider, once you've got this kind of setup, you can start looking and building up networks. And we're a few years behind, but we really want to push ahead to increase the numbers. So we've now got a new building. This is a 5-story building in our area, but there's a 3,500 square foot area for precision eye treatment, and that includes ASOs. So we're really looking to sort of treat the next set of patients, and we've already submitted the next mutation which is a family, but a single member, but there's also a family now who've been affected by this single mutation, which is less than 30 patients. So how many more patients can we treat? Well, we had a look at our database. So we've been very fortunate because we've had lots of eye testing prior to this trial. And we have 1,000 patients in our genetic database. So of those, 450 have been solved. That means we found a cause for that. And if you look at those numbers, then of those ones which are potentially treatable with ASOs, that's autosomal dominant disease and splicing abnormalities, make up about 40% of the solved cases. However, we're still limited by the N-of-1 trial. So the N-of-1 criteria at the moment is really less than 30 patients, obviously one patient, but less than 30 definitely. And of those, we're sort of thinking there's about 50 patients that we have. But that's 50 patients at one site. Now again, one of the benefits, as I mentioned, was that there's already a network of genetic specialists in eyes. So going across the states from all coasts, also the Midwest as well, there's some very big centers that have done their own testing. And also testing supported by a big charity called Foundation Fighting Blindness, which is another charity which does genetic testing. Their database has 20,000 patients, so I think probably 22 or 23,000 patients now. And that's just in the US. And again, that's not all the patients, because not everyone has had testing. So extrapolating our figures and looking at the numbers and the ratios, there's potentially 1,000 patients that maybe benefit from the N-of-1 style therapy. But really, well, one of the things that we should really be discussing is really going beyond the N-of-1 approach and looking at other approaches if possible. And that will obviously require FDA approval. Because if you could treat these patients, then potentially if you could treat the other patients with dominant diseases and splice site variants, you'd be getting up to 44,000 patients just with that database. So that's a huge number and a huge potential. So I just want to leave you with the fact that, eye side of things, obviously the diseases are not as severe systemically, but they still have major implications visually and also in terms of quality of life. The second thing is that the treatments are very easy, and the treatments have low burden in terms of the patient. And so I'd really like to explore increasing the amount of treatments for these patients. So with that, thank you for listening. Thank you especially to the family, some of who are here, and the patient. And special thanks to certain people such as Lisa Garone, who've really sort of sped up the drug development pathway in terms of the regulatory side, and lab members as well as clinical trial team. Again, as we talked about before, you need a village really to sort of be able to do this. So thank you again.